working for the crime lab. Ooh, crime lab. And we had to try and track down the perpetrator of the crime. Okay, so we've got our forensic sample here. And we're just doing a series of VNTRs, right? Because remember what we just finished saying a little bit ago is that every person is different, right? We've got a spectral karyotype of VNTRs that we can look at. Now, that is not the case if you have, I said this already, an identical twin, right? You are going to have exactly the same genotype as them. So, frame your twin. But otherwise, if you do this spectrum of VNTR analysis, you can actually amplify the DNA and get a very specific patterning of VNTR clones that you can then, again, run out on a gel after you've done all your amplification. And now, right, you've actually got 2 copies for each chromosomal marker that you're looking at. So, for instance, if you did 3 VNTRs, you'd have 6. Obviously, we're going to do a lot more than that, so we can really, really get a fingerprint, if you will. That's what this is called. Analysis of this person. This is DNA fingerprinting. Individual A, individual B, individual C, forensic sample. Which one's OJ? If the glove don't fit, you must commit. Yes. You must acquit, rather, not commit. Sorry. What was I saying? Boy, that populated our minds. Wow, very graduate school. It was a daily fun to go and watch TV for a little bit. Right? We can do this with VNTRs. We could do it with SNPs. We could do it with RFLPs. Obviously, the analysis would be different, right? If you were looking at a spectrum of SNPs, you'd have to actually sequence them to see whether the polymorphisms were there. Or if you were doing RFLPs, you'd have to do a southern to look for the different probes. So VNTR analysis is the fastest way to do that. But in theory, you could use SNPs or you could use RFLPs to do this same kind of thing. Each person's going to have an identity fingerprint of this type of polymorphic regions. Okay, good. Yay. So I want to spend just a couple of minutes on some very, very important VNTRs that we know actually cause diseases. Okay? So the first one we want to talk about is a polymorphism that's in the that causes Fragile X syndrome. Right? So Fragile X, as the name implies, is X-linked. So on the X chromosome, and you actually see this fragility occurring in the long arm of X at the very telomeric region, telomeric end, where the piece of DNA just gets really fragile and actually can break off. Okay? So there's very characteristic phenotypes, right? Long, narrow face and prominent ears. These children are very prone to having seizures, mental impairment, ranging just from learning disabilities all the way up to retardation. They're very prone to ADD and obsessive-compulsive disorders, right? Being an X-linked disease state, we have a much higher ratio of males that show the disease phenotype than females. It's the most common inherited cause of learning disabilities that we know of and that we can clearly define. And this is actually, and this is kind of, I was just reading about this in Science this morning, in fact, something I pulled out a couple of months ago, where this is actually a really, really unusual way that this silencing occurs. So what happens to this? Well, this is actually occurring in a gene called FMR1. So FMR1 is actually an RNA binding protein that normally is going to inhibit the translations of mRNAs that are specific in the brain that cause brain plasticity or neuroplasticity. And so normally you're going to inhibit that plasticity from occurring. So if FMR1 is not present, you get more plasticity in neural development, and they think that that may be linked to the development of some of these mental disabilities, okay, these learning disabilities potentially. So why does FMR1 not get expressed in these kids? And it's actually developmentally regulated, which is a really cool phenomenon. We're not going to get into that really. But it's expressed for a small amount of time during development, and then it gets turned off. So we would call that epigenetic silencing. But we'll learn more about epigenetic silencing next week. This is not the typical way that you would silence an epigenetic region. So what happens is the following. You have this VNTR region. I'm going to just draw this out. This is actually really kind of complicated. I want to make sure that you're following this. Let's see if we draw it over here. Okay, so we have a disease gene of interest, right? So this is, let's just call this as FMR1 here. Okay, so this is the gene, and we'll talk more about this next time. We're probably going to get to this, but this is the promoter region, right? So this is where the polymerase is going to sit down and actually start transcribing. Okay, so you're going to make a transcript, an RNA transcript, okay, that starts transcribing here, right? So the polymerase sits down, and we have an mRNA of a certain length. Now, this is the really crazy part of this. So, remember from your maybe a little bit of molecular biology before, the mRNA is not necessarily all coding sequence, right? So it's not necessarily all going to be translated, right? So the translation of the FMR1 gene actually starts somewhere down here. So this is translation starts. Okay? So this is where the coding sequence of the gene actually hit here. Okay? This region up here is a region that we call the 5' UTR, and we'll talk more about this again later. But UTR means untranslated region. Okay, so that's a part of the gene that gets transcribed into the RNA, but it does not get translated. Okay? So the crazy part about this actual VNTR is that the VNTR is encoded here in the 5' UTR. So it's a CGG repeat. CGG, 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 blah, 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 blah. And it goes on, and I think it has to expand. If you get, so there's a certain size that this VNTR has to be in order to get silencing occurring. And it's something like, if I remember correctly, 200 copies. If you get higher than 200 copies, you're going to get silencing occurring. If you're lower than 200 copies, not necessarily going to see silencing. Okay, so there's kind of this cutoff point where at some point it gets big enough that we start getting silencing. So the crazy part about this is that the mRNA comes back around and actually binds to the DNA where it's transcribed from. So the CGG sequence comes back around and hybridizes to the template strand of the DNA. So you get this RNA-DNA hybrid being formed in the region of the gene where the promoter sequence is and where the untranslated 5' sequence is. So the mRNA, as it's getting made, is coming back around, invading into the DNA, and actually causing the site to not be allowed to be transcribed anymore. So it's, in effect, silencing that area by having this hybrid formation. Really insane, very, very bizarre characteristic. You would not see this normally happening, right? Normally, your mRNAs are not coming back in and invading any strands. This is a very, very unique situation, and it has to do with the size of this repeat BNTR. So, right, it's not because of a protein problem. It's actually because the RNA is coming back around and it's recognizing this same sequence that's occurring in the DNA sequence and then hybridizing to it. Again, very, very unusual situation, but something that they figured out is actually happening in this hybridization. And it only occurs at certain times during development, right? So it starts happening at about 60 days post-neural differentiation. And they think that that has something to do with the helicase activity in this area, that certain helicases are present at certain times during development. Once those helicases are gone, this RNA can now slide in and invade the strand. It's really kind of, again, funk dog type of thing that's happening, but it's really pretty darn cool. And it only occurs, again, if this region is big enough. You have to be able to hybridize that RNA back onto this sequence that's large enough that you can have recognition occurring. Okay, so this is one way that we get disease states due to these VNTRs. Again, large majority are not disease state causing, but this one happens to be. Right, and it's not in the coding sequence. That's kind of the kicker. You'd think, oh, well, we're safe if it's not in the coding sequence. Well, it turns out in this situation, we're not safe from that. The other one that I want to talk a little bit about, and this is, I have a little bit about it in your notes section, is Huntington's disease, right? So Huntington's is another situation where you've got to get an expansion that is big enough. Okay, so up to about 35 repetitions, totally fine. You're not seeing any type of problems in the Huntington protein. However, as soon as you get past 35, you start having aggregation because this time the VNTR is actually in the coding sequence of the protein. And so it encodes for a glutamine, string of glutamines in the protein. As soon as it expands to a large enough tract of glutamines, you start getting agglutination, protein aggregation, of the strands of Huntington protein, and they essentially drop out, and you start getting aggregation and no function of that, or improper functioning of the Huntington gene product. Okay, so. The kind of crazy thing about Huntington's is that it expands as it's being inherited. Okay, so you can have, and it's generally a late onset problem, right? So you have a person who has, let's say, 30 or 32 or 33 repeats. They're okay, but as soon as you start getting up near that threshold where it starts being problematic, you actually get expansion of the repeat more frequently. Okay, so if I'm a dad, and it actually happens more on the parental strand paternal strand than it does on the maternal chromosome. So if I'm a dad and I've got a repeat region that's, say, 32, the likelihood that it expands to over 35 in the next generation is actually much higher. So as you get closer and closer, and again, this is a mutation that is inherited and it's expanding in the germline. So as you expand, as you get closer and closer to 35, it starts jumping and more frequently expanding to a problematic size as time goes on, as the inheritance pattern goes on. And we don't know why that is, but we know that it does occur, that as you get closer and closer, the more likely you are to have an expansion occurring in the next generation. And remember that next generation then inherits the disease probably sooner, so that you get an earlier onset phenotype in a next generation than you would in the parental generation. Does that make sense? Kind of funky again, but it is, with these VNTR expansion diseases, it's a threshold that you have to cross, and then after that, you've got problems occurring, whether you've got silencing or whether you've got problematic proteins that are being made. It doesn't really matter. You've got a threshold for expansion, and then you see problems. Yeah.
How is the late onset caused?
So it's an aggregation issue over time. You just start getting more and more aggregation of the protein. And that if it's got a bigger tract, it's going to aggregate more quickly and more easily. Okay, so not so good things, right? So that's the kind of problematic part about VNTRs. As we said, 99.9% of them not problematic at all because they're in non-coding sequence. However, these ones that cause disease states happen to be either in promoter regions where you're causing epigenetic silencing. In the case of Frax, it's in the 3' UTR of the mRNA, and in the case of Huntington's and several other polyglutamine tract diseases, it's actually in the coding sequence of the protein itself. So, multiple ways of having these problems in the VNTR situation. Okay, let's talk about this really cool technique that can be a gigantic can of worms. Okay, so microRNA analysis, and hopefully you guys have kind of heard this terminology. So, expand your mind this way. We've been talking about single genes, right? And single things that we're tracking, single VNTRs. What if we wanted to look globally? What if we wanted to look at the entire coding sequence of an individual, the entire expression cassette, if you will, of an individual to see whether or not there were changes in different states of time, right? So if there's, for instance, a cancer, a tumor that you've got, and you want to look at the expression of gene products in that tumor versus an area of the individual that doesn't seem to be tumorigenic, right? So a wild type area. You can look for differences in expression patterns of the mRNAs, or the genes of interest, by doing what we call microarray analysis. So this is really great for looking at comparative samples, right? Different tumors. Tumor versus wild type. Infected versus uninfected, right? This is what we do all the time, right? We're looking at expression patterns in infected cells versus uninfected cells to see what that virus is doing, for instance, or bacteria or anything, to the infected individual or the infected cells in terms of expression profiles, right? So microRNA analysis is all expression profiling, right? So you have to look at transcripts, mRNA transcripts, right? So if a gene isn't expressed, you're not going to get a transcript, right? So we can look at downregulation. Is that gene not being expressed now due to this new state of affairs? Is it upregulated? Are we getting more expression than we were in a normal state of affairs? Is it turning off? Is it turning on at different time points during infection, right? So you can do a time course of infection to see if it changes over time. You can look at analysis of different samples within a given individual. And that's actually a really powerful to look at. For instance, solid tumor sample versus normal blood sample, okay? And see what genes are being expressed. So how do you do this, right? This is a transcript analysis. Transcriptomics, if you will. We love to use omics now in molecular biology. Everything is omics. So transcripts. What do we do with transcripts? We don't like transcripts, right? We like RNA. We don't like RNA. We like DNA. So what are we going to do with our transcripts, or our RNA that we've isolated from these peeps, or from these different samples? We're going to reverse transcribe it. Bam, right off the bat, we're just going to make cDNA, okay? Because cDNA, as we know, is much, much more stable, right? RNAs are super labile because they're single-stranded molecules. So we're going to extract our RNA out of our two samples. Here we've got a normal and a tumor sample. We're going to extract the RNA. Cool thing about this is, as we're making our cDNA of interest, we're going to use labeled nucleotides so that we have, let's say, green fluorescently labeled nucleotides in our normal sample and red fluorescently labeled nucleotides in our tumor sample, okay? Why would we do this? We want to tell them apart, right? We want to be able to tell them apart. We want to be able to tell normal versus tumor. So, right now remember, everybody think about our reverse transcription, right? We're doing the process. We've now made our first strand synthesis, second strand synthesis. We've got our cDNAs. Now we've got this whole big panel of cDNAs that we've got, okay? One's labeled with green, one labeled with red. Smack them together, right? Equal parts. Tumor, normal. We can mix them together because they're all equally labeled in terms of their cDNA content. Right? Each strand of cDNA is labeled. Okay? So now we do that. And we hybridize them to what we call a cDNA array, a chip, if you will. And across this chip is arrayed every single gene of interest. Our 21,000 genes are all arrayed on this chromosome. So cDNAs for each of our arrays, each of our genes are on here. Now we've got multiple samples from each gene on this array. Because, right, we're not going to put the whole damn cDNA on there, right? We're going to put fragments of it. And we're going to want to have multiple copies of each gene. Why would you think that you want to have multiple copies? There's enough work as it is. Pardon? In case something happens, in case one doesn't seem to hybridize all that well, right? Some funky thing with that chip. I'm going to get back at that manufacturer, screw it on that spot, right? But you don't want to just have one spot for each gene, right? That's asking for trouble when you just have one of anything, right? You always want to have multiple spots that we have for the same gene because, right, what happens if that piece of cDNA from that gene happens to be like super GC rich, right? We didn't get a really good amplification on that. Or it doesn't hybridize very well at the exact temperature that we're doing this hybridization at. We don't want to have and rely on saying that gene of interest was upregulated or downregulated from one spot, right? So we do what we call an analysis across that gene, right? So if we have 6 spots for any given gene product, well, we damn well better have at least 4 out of the 6 showing the same pattern of upregulation or downregulation. If we got a 50-50 change, throw it out. That doesn't mean anything, right? So we're looking for a consensus. This gene's going up, this gene's going down, this gene's staying the same, okay? So that's exactly what this is right here, okay? So we've mixed our two probes together, right? Red, green. What does red and green make on a color palette? It does. In fact, it does. Right? Think about your color wheel. Red and green actually makes yellow when they have equal parts. Now you can have gradations of red or gradations of green, depending upon how much you've got of any given individual color. Okay? So if you've got exactly the same amount of that cDNA in your tumor and your wild type cell, they are going to mix together, they're going to hybridize to that spot at the same exact ratio, and you're going to have a yellow spot. If you've got more normal cDNA, you're going to get a green spot. If you got more tumor cDNA, you're going to get a red-ish spot, right? That's going to be ish, depending upon how much higher the amplification is in one or the other, right? So if it's super red, and there doesn't seem to be any hint of green there, well then that probably means that that gene's actually been turned on in the tumor, right? So normally your cDNA would not be seen. You don't have that gene expression in a normal phenotype. However, if it's blazing red, you know, bam, you've got turned on expression of that given gene, right? That's what I mean by these gradations. It may be that you've got a little bit of green, but you got a whole lot more red, then it's not going to look exactly red, because there's going to be a little bit of green in that. So there's this gradation of green versus red, and all the way in between to yellow. Does that make sense? That's how we get our fold differentiation, right? So if there's, let's say, 5 copies of green and 25 copies of red hybridized to a given spot, well, then we've got a 5-fold differential, 5 versus 25 copies. We got 5-fold more of our mutant, for instance, our tumor DNA in that spot. Does that make sense to everybody? Gradations are a little bit funky, but let's just say, we can even just think about it as plus minus, right? So if it's red, it's tumor DNA. If it's green, it's normal DNA. That's actually being expressed there. And so we can look at these and say, well, in this tumor, cyclins seem to be upregulated. Or in this tumor, P53 seems to be knocked out. Or RB seems to be knocked out. Something that drives the cell cycle should be upregulated in the tumor. Something that stops the cell cycle should be downregulated in the tumor, right? And we'll talk much, much more about that when we talk about the cell cycle regulation genes and who's where. But that's the kind of thing that we're looking at, right? When we're looking at expression profiles to see what things, for instance, make a tumor, right? So we talked a little bit about that the first day. We can screen and look at microRNA analysis, or excuse me, microanalysis for 25 different breast cancers, okay? We can look at the expression profile for those. We can look at 25 different colon cancers. We can look at 25 different lung cancers. We can see whether or not there's expression profiles that are specific for breast cancers versus colon cancers versus lung cancers, and try and see whether there are genes that are upregulated or downregulated more likely in a given type of cancer versus another type of cancer. So there's a beautiful analysis that was done recently looking at, since breast cancer is our favorite problematic thing this year, looking at, right, so you guys have heard maybe of what we call triple resistant breast cancer lines. So triple resistant breast cancers are ones that are not expressing the 3 receptor molecules that seem to be problematic in breast cancer. So they're They're triple negative for the EGF receptor, they're triple negative for the FGF receptor, and they're triple negative for the progesterone receptor. Oh, sorry, estrogen, progesterone, and HER2, right? HER2 is the other gene receptor that they're negative for. So the reason that they look at those 3 receptors is because oftentimes in breast cancers, those 3 receptors are very highly upregulated, and they drive the cycling of those cells and the metastasis of those cells. And so when you have a tumor that's triple negative that doesn't have any of those 3, you're sitting there saying, well, what the hell? Why are these things metastasizing? They don't have any of the same markers that would normally cause this to metastasize. So they've actually looked and done arrays of triple negative tumors versus just kind of run-of-the-mill breast cancers, okay, tumors. And what they've found is they've now localized a set of G protein coupled receptors that are on the surface of the cell that seem to be susceptible in these triple negative people, that they're upregulated highly in the women that have triple negative tumors. And what's really cool is they've taken this one G protein coupled receptor and overexpressed it in tissue culture, right? So now we take the cDNA, pop it into a cloning vector, express it in our cells, overexpress it, and bing, bang, boom, those cells are cycling like crazy, and they're metastasizing, they're moving, they're invading really fast. So good target for us to be trying to say, okay, how do we turn off that expression in these women that have triple negative breast cancers, right? Because oftentimes, and this is, the triple negative cancers are the ones that are the most aggressive. They're very, very metastatic, and they have a very bad prognosis. And so if we can target those and get some therapies targeted for downregulating expression of this GPCR, it might actually be really, really beneficial to these women. So cool, right? That's a really cool new thing that we found. Okay. That's microRNAs. Bloody, bloody, bloody. That's all we have time for today. So on Friday, we're going to just touch for like 10 minutes on deep sequencing to just get a taste of that and how crazy it is. And then we're going to do transcription. And we're going to get all the way through transcription on Friday, because we have to. Because we will. Yeah, great. So the